So, can organic carbon be produced abiotically under biologically relevant conditions? As discussed before, Fischer-Tropsch type synthesis reactions are catalyzed by various metals such as nickel and iron. At temperatures between 0 and 100 degrees, and at pressures similar to that of Earth at sea level, iron can't oxidize hydrogen, and nickel can't reduce carbon. Iron can reduce carbon, and nickel can oxidize hydrogen, but they can only do so under certain pHs, with nickel oxidizing hydrogen under alkaline conditions, and iron reducing carbon at neutral or acidic pHs. When the nickel and iron are in a cluster, electrons stripped from hydrogen can be shuffled back and forth between nickel and iron, and thus can be used to reduce bicarbonate to formate. It has been experimentally demonstrated that when alkaline hydrogen-rich water is separated from acidic carbon-rich water by a semiconducting barrier, it will result in the production of organic carbon. So, a variable pH environment is needed to abiotically produce organic carbon under biologically relevant conditions, and the cross-membrane pH gradient is a near universal feature of life. But could such an environment have existed on the prebiotic Earth? Modern serpentinizing systems exist deep in the ocean or far underground, and thus have a relatively constant pH. However, 4 billion years ago, the surface rock was far more mafic than it is now, implying that serpentinizing pools would have been prevalent on the surface. The high concentration of carbon dioxide in the atmosphere would cause the rain to have a pH close to 6, which would then leach calcium out of the rocks as it flowed along the surface. As this water poured into serpentinizing systems, the alkaline pH would cause the calcium and carbon dioxide to precipitate out as calcium carbonate, forming an intricate labyrinth-like structure. Depending on the weather conditions, acidic carbon-rich rainwater and the alkaline hydrogen-rich vent water could be on opposite sides of a calcium carbonate barrier. If there was an iron-nickel-sulfur mineral inclusion, the abiotic production of organic carbon would take place. The rate of organic carbon production would be greater than in other hydrothermal systems, and it could become sufficiently concentrated to initiate pre-metabolic pathways to facilitate the transition from simple organic molecules to the first biomonomers. One pathway, which is nearly ubiquitous across all branches of the tree of life, is the TCA cycle. In our cells, acetyl-CoA is combined with oxaloacetate to form tricarboxylic acid, hence the name TCA cycle. And, through a series of reactions, carrier molecules take electrons off the carbon and shuttle it to oxygen molecules producing water, and 2 carbon atoms are removed as carbon dioxide, regenerating oxaloacetate, which can then react with another acetyl-CoA for another turn of the cycle. We, as aerobic organisms, use this process to produce energy. However, some bacteria can run it backwards as their way to uptake carbon. Here, instead of being run oxidatively or clockwise, it's run reductively or counterclockwise, where the aforementioned carrier molecules provide the organic molecules in the cycle with electrons. Here, 2 carbon dioxide molecules are taken up and an acetyl-CoA is produced, which can then be fed into biosynthesis pathways. But virtually all organisms which use the TCA cycle use intermediates known as alpha keto acids as precursors to the production of amino acids. Similar abiotic reaction networks have been observed being catalyzed by iron-sulfur minerals and will occur in cyanosulfidic conditions. The succinate dehydrogenase enzyme used in the TCA cycle possesses an iron-sulfur cluster. The direction of the reaction would depend on the availability of oxidants or reductants in the environment, running clockwise in a more oxidizing environment and counterclockwise in a more reducing environment. These oxidants and reductants can be inorganic, such as molecular hydrogen and perchlorates, or be highly reduced or highly oxidized organic molecules. In order to run, the abiotic analogs that have been observed thus far have to be fed organic molecules such as acetate and glyoxylate. Some of the steps remove carbon atoms of organic molecules as carbon dioxide, but as of now, no abiotic TCA cycle analog has been observed binding carbon dioxide to organic molecules, although mechanisms have been proposed by which this could happen. So, while the abiotic TCA cycle analogs could provide early life with the precursors of amino acids, it's unlikely it could have been used as a means to assimilate carbon dioxide. Ancient serpentinizing mud volcanoes from 3.8 billion years ago were replete with ammonia, and while there's still debate as to how it was produced, the alpha keto acids produced by the abiotic TCA cycle analogs could be reacted with ammonia to form amino acids. Two other metabolic pathways that are common across all branches of the tree of life are glycolysis and the pentose phosphate pathway, or the PPP. Being multicellular organisms, we often think of these pathways as means by which sugars are broken down into simple organic molecules to produce energy. However, these pathways can be reversed to produce sugars from simple organic precursors. To be fed into these pathways, sugars will have a phosphate group bound to them, and the enzymes that catalyze the reactions in the pathway have magnesium cofactors which interact with the phosphate group. Abiotic analogs of glycolysis and PPP have been observed in chemical conditions similar to the oceans of 4 billion years ago. In the abiotic reactions, ferrous iron ions perform a similar function to the magnesium cofactors in biological systems, but it should be mentioned that terrestrial serpentinizing systems would have had both magnesium and ferrous iron ions. In an alkaline environment, the reaction networks more closely resemble that of the pentose phosphate pathway, while in acidic or neutral conditions, it more closely resembles glycolysis. However, like their biotic counterparts, the abiotic analogs of PPP and glycolysis require phosphorylated organic molecules to proceed. In modern organisms, this is provided by an adenosine triphosphate molecules, but these were likely not around when life began. Much of life's biochemistry is powered by phosphorus, where a biomolecule is activated by binding a phosphate group, making it more reactive. Assembling biomolecules from simple organic precursors decreases the entropy of a system. So, in order for the delta G to be negative, high energy bonds, such as those between phosphorus and organic carbon, have to be broken. Most phosphorus in the environment is in the form of orthophosphate, and this is highly inert and will rarely react with biomolecules to form organophosphates. Usually, the production of organophosphates involves cleaving a phosphodiester bond which binded two phosphorus atoms in a polyphosphate group. In living organisms, this bond is usually the one between the second and third phosphorus groups on an ATP molecule. In order to replenish ATP from ADP and orthophosphate, an input of energy and complex molecular machinery are required, machinery that would not have been available on the prebiotic Earth. However, there is an iron-nickel phosphide mineral known as schreibersite, which, while rare on the surface of Earth, is abundant in metallic meteorites. Given that the origin of life occurred in the context of the late heavy bombardment, meteorites would have fallen regularly. When schreibersite is in water, it corrodes to form reactive phosphorus species, or RPS, such as pyrophosphate, metaphosphate, and phosphite. These can then react with organic molecules to form organophosphates. For example, pyrophosphate, depicted here, will react with an alcohol group to form an organophosphate, in so doing cleaving this phosphodiester bond and releasing an orthophosphate. This organic molecule can then be fed into prebiotic synthesis pathways. Now that we've established mechanisms by which the building blocks of life could be produced on the prebiotic Earth, we can switch gears a bit and talk about the origin of replication. All living organisms reproduce and in so doing convey genetic information from one generation to the next. This information is carried via the DNA molecules which specify how the organism will produce proteins. As discussed before, enzymes are a variety of protein that allow metabolism to proceed. In order for information in a DNA sequence to be translated into an amino acid sequence to make a protein, it is first transcribed into an RNA molecule via a similar mechanism to replication. However, DNA requires a complex system of molecular machinery to be replicated, transcribed, and translated, all of which, again, would not have been present on the prebiotic Earth. However, it has been speculated that the first organisms used RNA as both genetic material and as a catalyst for metabolic reactions. Here, we'll explore this possibility. Enzymes catalyze the majority of metabolic reactions in living organisms, and they do so via interactions between the variable R groups and the substrate. However, in the 1970s, it was discovered that RNA sequences can have catalytic activity as well, and these sequences are known as ribozymes. RNA, like DNA, is composed of many nucleotides, and each nucleotide is made up of a ribose sugar with a nitrogenous base on the first carbon and a phosphate group on the fifth carbon. When joined in a sequence, the nucleotide will join the phosphate group to the third carbon of the adjacent nucleotide. To this day, some RNA sequences are still used by cells to catalyze reactions, such as the ribosome, which translates messenger RNA sequences into proteins. In order to get RNA polymers, we first have to have individual RNA nucleotides. For a long time, it had been known that boiling aldehyde can produce sugars, and boiling simple nitrogenous compounds such as cyanide, cyanocitylene, and urea will produce nitrogenous bases. The problem with this mechanism is that while it will produce ribose and the nitrogenous bases used in RNA, namely adenine, cytosine, guanine, and uracil, it will also produce many other sugars and nitrogenous bases which can't be utilized by RNA and would inhibit its activity. So, what is needed is a process that will primarily produce ribose and the aforementioned four nitrogenous bases. The abiotic analog of the pentose phosphate pathway would produce ribose. Aldemerization of glycoaldehyde phosphate and formaldehyde will also produce ribose, but the phosphates will be on the second and fourth carbon. Figuring out how the nitrogenous bases were attached to the ribose has proven to be difficult, because biochemists couldn't get the glycosidic bond which binds it to the ribose to form abiotically. Instead, it is now believed that the nitrogenous bases were synthesized on the ribose itself. This process would start by a cyanamide molecule binding to the first carbon, forming an amino oxazole. Then, other simple nitrogenous compounds would be added to form the nitrogenous base. An added benefit of this process is that the amino oxazole will crystallize out of solution when drying, essentially purifying the ribose. The last thing that has to be accomplished is the binding of the phosphate to the ribose. It has been shown that borate can catalyze this reaction, and it may be that it could bind a pyrophosphate group derived from schreibersite corrosion to the fifth carbon, but this has yet to be experimentally demonstrated. Borate can also facilitate the binding of urea to the first carbon, which the nitrogenous base can then be built around.